tucked in.
So, I don't know, he may be overwhelmed before long.
There you go.
Need some good stuff. You just need that.
4 weeks in
We're not tight today. Whoa!
I think I'd rather get my ankle replaced than my quad tendon repaired. To tell you the truth, I think I'd rather get it than anything else. I'm still struggling with this. The question is, will I ski with an ice or without it? That's going to be my only question. I find out in 2 weeks.
You don't like it?
Yeah, I skied right until I died. Right up until Friday the 13th. And that was the end of it.
Is that what it was?
Just an error in judgment. Recovery. No, I was coming back. It was really heavy snow. But when it was uncut, it was okay. So I came back full time hole. I thought I'll just take one. It was all cut up. One ski in the snow and one ski in the air. Then I felt this really bad pop and I just was really loud snap and I couldn't stand up. It's like, I'm screwed. So, but I had to call myself on the radio. I got a 64-year-old male with a knee injury. It's time hole.
He's in bad shape.
Where's the patient? I'm about 70 meters north of Tower 6. Then they get down, they go, where's the patient? I said, I'm the patient. What? Yeah, I just tore my ACL, which is what I thought.
We were with my dad and my uncle were skiing, and he went off and jumped, and he hit. You could tell he drove. So my dad helped him down. It was the beginning of the day, first run. He's like, for my uncle, my dad went out skiing the rest of the day. He said, I was going to ski in the lodge, but I dedicated my day to skiing. I dedicated the day to skiing.
Is he coming today or is he gone again?
You know better than me. I don't know where they are.
I don't know anything.
You're like me. You're out of town.
Yeah, well, it's about time. I mean, they had that scare with your mom, what, 10 years ago?
That was like 15 years ago.
So she's obviously...
Yeah, they're enjoying life.
Fortunately, they're reevaluated.
It did change things for them.
So have you figured out your schedule out there? Are you still seeing 7 days an hour?
It's hit or miss. Sometimes it's nobody.
I have days like that too. Sometimes it's nobody.
7 or nobody.
It's like,
It's really a feast or famine.
how far behind am I? I haven't even started yet, and I'm at work. How does that work? So no lunch. Out there on vacation. See, fishing or working?
Oh, that one.
I still do that.
I know. It's like we're calling out.
Yeah, talk to us. All right, Len's out of town, so he put me in charge.
All right.
He's from Alaska, not Hawaii, for once. But we have Dr. Choy coming to talk to us about asthma biomarkers, so welcome.
Good morning, everyone. Can you hear me okay? All right. If my voice trails off towards the end of the morning, please let me know and I'll speak up. So once again, I'm Dave Choy from Genentech. First of all, I want to thank everyone here today for listening here, and I'm really happy to share some of our work. As you can see from the title, I'm going to be talking about biomarkers. The focus of it will be around biomarkers for Omalizumab. So really kind of what I hope to share with you is the perspective from the drug discovery and development perspective of what we learned from studies of Omalizumab, biomarker-enabled studies, to enable decision-making for other pipeline therapies. These are my disclosures. Okay, so first of all, the first question, why study biomarkers for Omalizumab, a market therapeutic for severe asthma? So there's a lot of different motivations for why we might want to do this, but in particular, my motivation is really to facilitate the interpretation of the clinical activities of novel therapies, especially if you're not in the business of trying to develop new therapies that are more effective, that can ameliorate symptoms in a larger population of asthmatics. And also something that is of particular interest to me, it helps to enable this idea of reverse translational research, where we've invested in studies in thousands of severe asthmatics, where we characterize them on a molecular level, and how this provides us insights as in terms of new therapies and as in terms of patient heterogeneity, in which we believe there's a great deal of patient heterogeneity in the context of asthma. So as in terms of our agenda today, what I would like to walk through with you today is a little bit about the past, and I think you're familiar with a lot of this data, but I think to kind of reintroduce some of the data of Omalizumab with respect to its biomarkers And hopefully I can provide a little bit of color as in terms of the perspective about how we've interpreted and utilized this data. And in the recent past, how we utilize this information for decision making for similar MOAs such as Omalizumab, and I'll provide a little bit of an introduction as in terms of what Omalizumab is to you later on in the talk. And finally, some unpublished data that I'm really excited about is as in terms of our study about the pharmacogenetics of Omalizumab. It's really kind of the discovery of genetic predictors of response to Omalizumab. And in itself, I think it's actually interesting and useful for, and there's utility for Omalizumab, but I think it actually has some implications for new therapies as in terms of targeting different aspects of mast cell biology. And then finally, applications. Okay, so to kind of really introduce you to the concept of reverse translational research, and I think, several people have different perspectives on this, and so I feel that I'm really lucky working at Genentech, as I had mentioned, that we've invested in studies and attempts to develop and discover new therapies for asthma for quite a while now. And what this has really resulted in is that we have tens of thousands of patient-level data, samples, that we're continually doing research on. And really the goal is to utilize these clinical trial-defined outcomes and biomarker-defined groups to inform earlier pipeline programs. And so some of the things that we're actively doing is we're doing whole genome sequencing in our Genentech cohorts, and currently there's over 5,000 severe asthma subjects that are well characterized clinically, where we have whole genome sequencing data. We retrospectively have executed analyses of non-invasive biomarkers for exploratory purposes to have a better understanding about the pathway activation in these subjects and its heterogeneity. And some of the questions that I've been interested in, but I'm not going to be talking about today, is how it relates to extrinsic factors, which some may argue is actually intrinsic factors, such as the airway microbiome, and also looking at the molecular phenotypes, such as looking at the transcriptome of the affected airways. So I just want to walk you through what our working model is as in terms of severe asthma. It probably bears a lot of similarities of how other people are thinking about it. But our approach as in terms of developing and identifying new targets is to try and have a better understanding about the relationship between environmental or extrinsic triggers to the biological pathways that we intend to target to ameliorate asthmatic disease and the resulting tissue pathology that is observable. And with respect to clinical outcomes, things that we can actually measure in a clinical study, and some of these outcomes that we think actually provides meaningful clinical benefit to patients. As in terms of the extrinsic factors, we have a greater appreciation that it's just not an intrinsic disease, and that normally innocuous stimuli, such as allergens, viruses, bacteria, fungi, pollutants, etc., these elicit symptoms in asthmatics that are normally in a healthy patient, usually normally be innocuous. So as in terms of the biological pathways that we think are relevant, I think what has been most characterized is really this type 2 high phenotype, and so I'll discuss that a little bit more in a subsequent slide. And really, what I'm trying to communicate is that we believe that it is heterogeneous with respect to these biological pathways that we believe are active in severe asthma. As in terms of the nature of the inflammation, as in terms of type 2 high, typically it's associated with these type 2 cells that can include Th2 cells, ILC2s, even eosinophils. And really they're defined by their patterns of expression of cytokines, including OB4, 5, and 13. Among the type 2 high spectrum, there's also another phenotype that we described before as this complex phenotype of mixed inflammation, where we see among subjects who are predominantly eosinophilic, they also have measurements as in terms of type 17 signaling, as in terms of their disease. And this also results in complex granulocytic inflammation, including neutrophils. Furthermore, what we think is a little bit more independent of type 2 inflammation, yet can also be activated by type 2 inflammation is mast cell biology. And we think that this is something that's been very relevant as in terms of the MOA of Omalizumab, and it's a big focus for new therapeutics as in terms of us going forward, because we believe that mast cell involvement is the determinant of asthma. And as you can imagine, there's a number of soluble secreted mediators, such as proteases and histamine, that might be involved as in terms of the pathophysiology of asthma. And furthermore, another focus at Genentech is neuroinflammatory pathways that we think are relatively type 2 independent. And so this is the area that we think is of a great unmet medical need and so great focus at Genentech. All of these pathways can culminate in a complex fashion as in terms of tissue remodeling and the resulting pathology of the airways, where in the most severe cases of asthma, we have indications of fibrosis, edema, and also hypoplastic airway smooth muscle cells, and also differentiation of ciliate epithelial cells to secretory cells, where it's believed that in the most severe cases there's pathologic mucus. And these all result in clinical outcomes that we can actually measure in the clinic, as in terms of acute worsening of disease, such as exacerbations and airway obstruction and airway hyperactivity and symptoms. So I think for this crowd, I don't really have to go into depth as in terms of the clinical outcomes, but I just want to make a few points. As in terms of the clinical outcomes we primarily capture in our studies, these are really the cardinal clinical features of asthma, episodic airway obstructions. So when looking at FEV1, while we think that as in terms of having a clinical effect on this outcome may be of minimal clinical benefit to patients, it's a very sensitive measure as in terms of getting an idea of drug activity of a molecule early in clinical development. And the way that we link it to is that it's a direct measurement of the airway caliber, and this can be affected by airway smooth muscle tone, inflammation, and mucus. Something that we think that's really important as in terms of the morbidity of the patients are exacerbations. And this is for many of our studies, the approvable endpoint where we hope and aim to make significant improvements as in terms of exacerbations. And the point that I really want to make about this is the greater appreciation about the heterogeneity of instigators of exacerbations and how we think that these might be ameliorated by targeted therapies, which I'm going to go into a little bit more. So, as in terms of the variability or the heterogeneity of exacerbations, what I want to draw your attention to is this epidemiologic study, and this was based on electronic medical records over approximately a 10-year period from a Canadian hospital system. What you see on the x-axis are the weeks of the year, so we have approximately where I've annotated September here, and on the y-axis we have the multiple weekly mean of hospitalizations due to their asthma or COPD for these particular age strata that you can see in the legend. And hopefully what you can notice, and I think you can appreciate, is that there's seasonal variability. And it's largely, while they have not shown definitively that viral infection is the actual cause, it's been shown to be correlated, at least, with respect to rhinovirus prevalence. Rhinovirus being a much more prevalent virus, building up the hypothesis that there must be, or there could be some sort of interaction between viral triggers and acute worsening of asthma symptoms resulting in exacerbations. So how does this really affect how we think about targeted therapies? Now I want to draw your attention to these two particular studies on the upper left that you might be familiar with. One of them is the ICATA study, which was a study of omalizumab in mostly adolescents, severe asthma subjects. And what you're looking on the y-axis, percent exacerbations per calendar month. If you look at the lower plot, this was a study of lebrikizumab, which is anti-IL-13 in adults with severe asthma. And what you're looking at on the x-axis, because of the global study, is hemisphere normalized month. And on the y-axis, we have per month exacerbation rate. Hopefully what you can appreciate is there's a very, very distinct similarity as in terms of the seasonal pattern, both in the placebo arm and as in terms of the efficacy on the on-study drug participants. And I want to point your attention to two different things, is that for both studies, we see an increase as in terms of the exacerbation rate in the spring, followed by the nadir in the summer, where it essentially overlaps with the exacerbation rates overlaps with on-study treatment for either omalizumab or lebrikizumab. Also, in the fall, we see the expected increase, and this is largely attributed to the return of school for children, where we have a community of active infections leading to exacerbations. And correspondingly, we see a minimal treatment effect in the summer, and we see the elevated treatment effects both in the fall and in the spring. So if you were to accept that there's different types of exacerbations, and different types of exacerbations were amenable to different targeted therapies, perhaps we could think about it this way, where we think in A, that mechanistically, that these are allergic mechanisms that are primarily driving their exacerbations, that we could think that there is an increase in air allergen exposures in which patients have been sensitized to, and through IgE, Fc epsilon receptor mediated degranulation of basophils and mast cells in the airway leads to inflammation or cytokine-driven inflammation in the airways, leading to exacerbation of disease. It's a little bit more complex in how we think about the efficacy of omalizumab with respect to potentially a viral trigger. And I think a lot of the work from Patrick Holt has provided some insights that might mechanistically explain this. So if you think about exposure of rhinovirus to the airway epithelium, leading to upregulation of type 1 interferons, what Patrick Holt and others have shown is that airway mucosal dendritic cells upregulate Fc epsilon receptor. And if you think about the atopic airways, where there's a de novo synthesis of IgE and there's air allergen, this could potentially lead to a cross-linking of Fc epsilon receptor on the airway mucosal dendritic cells, where it's been shown in vitro to lead to de novo cytokine production, the release of other mediators, which may lead to intense inflammation leading to exacerbation. So it's thought that this is one of the mechanisms in which anti-IgEs or omalizumab might be efficacious for viral-driven exacerbations. Also, we believe that lebrikizumab may have been effective in this particular case, that it blocked the downstream IL-13-dependent inflammation in subjects. And then finally, to be honest, we have very little understanding about these other exacerbations that may occur at other times of the year, and there's been other hypotheses in the field. So some of the hypotheses that have been driven into the field and have been described by Chris Breitling et al. And so what you're looking at is really kind of a summary of a culmination of the work that they've done. And what Chris has done is that he's followed asthma subjects and COPD subjects, and he's studied them at the time of exacerbation, where he's done intensive biomarker analysis as in terms of sputum samples, peripheral blood, and also assessed them clinically. And to summarize a lot of the work that he's done is that he's defined distinct clusters of phenotypes of exacerbation in terms of asthma subjects and COPD patients, where you can see cluster 1, they are primarily neutrophilic and they're bacterial, and they're associated with an increase of culture bacteria and also an increase of innate inflammatory cytokine signaling. If you look at cluster 2, this is predominantly what we think of as a type 2 phenotype, whereas in terms of granulocytic inflammation, it's dominated by eosinophils in the sputum, and as well as upregulation of cytokine mediators such as IL-5. And then finally, cluster 3, where by sputum and nasal aspirate confirmation of viral PCR positivity, that he sees another cluster where also coincident with increase of type 1 sputum mediators such as CXCL10. So this kind of sets up a framework that there is different types of exacerbations. Whether or not they're actually caught at a slightly different point in time temporally is yet to be resolved, but within 24 hours of displaying symptoms, these are the phenotypes he's described. So now I want to take it back a little bit to the way that we're thinking about biomarkers. And I mentioned to you that type 2 inflammation is heterogeneous in asthma, and the way that we define type 2 inflammation is really the nature of the inflammation that is driven by predominantly the cytokines IL-4, 5, and 13. And this could be produced by a number of cell types as indicated here. And the way that we think about biomarkers, in this case, blood eosinophils, serum periostin, and FeNO, so fractional exhaled nitric oxide, are a direct reflection of the activity of these cytokines. As many of you may appreciate, IL-5 is an important factor as in terms of eosinophilisis, and in conjunction with other chemokines leads to the recruitment of the eosinophils to the affected airways. IL-4 and 13 signaling through the IL-4 receptor system leads to a transcriptional profile of the structural cells of regulation of periostin, as well as NOX2, which is important in the production of NO, and we can measure that non-invasively by FeNO. With periostin, we believe that it's secreted basolaterally from epithelial cells, and it is able to make its way to the vascular plexus, where it can be measured non-invasively in the serum. As in terms of the activity of predominantly OFF4, as well as other activating molecules, leads to activation differentiation of naive B cells to soluble IgE-producing B cells. And as you can recognize, IgE decorates itself on the surface of mast cells in the presence of cognate antigen, allergen, and lead to degranulation and exacerbation of disease. So some of the therapeutic molecules that are under investigation at some point, or with respect to Omalizumab, an approved therapeutic, is kind of represented as in terms of where we think they are relevant in terms of this scheme here. So Omalizumab, which I'm going to be talking about later in a little bit more detail, targets a membrane-bound form of IgE B cells. So getting back to the original scheme, how do we think these biomarkers relates to the pathophysiologic phenotype in the airway? So what you're looking at here is a walk analysis from the Bobcat study. So it was a bronchoscopic study in which our aim was to really understand the relationship of non-invasive biomarkers with airway pathology. And so this is a slight deviation from how we reported this study, but the field has kind of coalesced to the idea of this 2 or 3% threshold of sputum eosinophilia to define eosinophilic airway inflammation. And based on this one particular cutoff, which in general is arbitrary as this is continuously distributed, so that we can see that all three, serum periostin, pheno, and blood eosinophil count, with high sensitivity and specificity, seems to identify eosinophilic inflammation, at least with respect to how this is defined. So getting at a little bit to the reverse translational research of how we can leverage our internal data, what you're looking at is a meta-analysis of probably 9 to 10 severe asthma studies. And if you're not familiar with this type of plot, it's a pairwise scatter plot where on the diagonal axis defines the variables of interest and their transformation. And the lower left references the scatter plot and is defined by the intersection of the diagonal as in terms of, you can see on this plot, the y-axis is serum periostin, the x-axis is serum periostin, the y-axis is blood eosinophils. And the upper right plots indicate their Spearman-Rose correlation strain. So hopefully what you can see here is that for a large number of subjects, we can detect a moderate correlation that is statistically significant. And this is kind of in line how we're thinking about it, as we believe that the type 2 cytokines, that are co-regulated in many cell types, and that these biomarkers are a reflection of that activity. Also, what I want to point out to you is the distribution of these biomarkers as well, is that all these biomarkers, if they are good indicators of type 2 inflammation in the airway, suggest that it's a continuous phenotype, that we don't have a dichotomous phenotype of a Th2 high subject versus a Th2 low subject. It's really a spectrum of type 2 inflammation that we see. While we don't have as much data on that, we see that with invasive measures of type 2 inflammation as well, such as eosinophils in the tissue or eosinophils in the sputum. So this is some data that you might be familiar with. This is data from the EXTRA study that was published a number of years ago. And I replotted this because I wanted to point out a few different things. And I just want to orient you as in terms of orient you to these graphs. So we have 3 biomarkers, serum periostin, pheno, blood eosinophils, that were measured at baseline in the EXTRA study. And the EXTRA study, for those of you who aren't familiar with it, was a study of Omalizumab in approximately 850 subjects, randomized 1-to-1. And we dichotomized the biomarker-defined population around the observed median, since kind of getting to the point that I made earlier, that we believe that these are a continuous phenotype. And really, to enable this analysis, we chose equal-sized groups. And when you're looking at the y-axis, is the adjusted annualized exacerbation rate. And if I can focus your attention to pheno, as I think it's an exemplar case, we see increased treatment benefit in the subjects who had high levels of pheno at baseline, and there's similarities with subjects with high levels of serum periostin and blood eosinophils. But the point that I wanted to make that really wasn't an emphasis as in terms of our Blue Journal paper is most of this treatment effect is driven by the prognostic effect. If you take a look at the placebo arms, these are the subjects that had more on-study exacerbations than on pheno-low subjects. And that it seems that Omalizumab, that one of its mechanisms of action was blunting this increase in exacerbations associated with the type 2 high phenotype, which kind of goes in line with our observations based on the seasonal analysis. So now I kind of want to shift your attention to some other data that you might be familiar with. What are some of the pharmacodynamic effects of omalizumab? And I think to kind of summarize them and pretend to some of the data that I want to show you is that it has a resulting effect on measurements of eosinophilic inflammation. As what you can see here on an earlier study is that they observed that there's a reduction in the sputum eosinophils as well as tissue eosinophils. And one of the thoughts is that the mechanism of action, which I thought was a very important aspect, is that it's in terms of omalizumab, the reduction of free IgE leads to the reduction of Fc epsilon receptor expression. So we think primarily this is one of the mechanisms of action of omalizumab. So this is an inhaled allergen challenge study that you might be familiar with, and it goes back a little bit farther back from John Fahey at UCSF. And the way that we've been kind of thinking about the allergen challenge is also a biomarker as well. Typically, an inhaled allergen challenge study for mechanisms of action that are ostensibly of allergic nature have been very useful as in terms of clinical development, but I see this really as a biomarker. And if you take a look at this inhaled allergen challenge study, you can see that omalizumab was effective as in terms of the EAR and the LAR, so directly affecting the allergen's ability to affect mast cell degranulation and also de novo cytokine synthesis. But also what they noticed in this study was there was a reduction of sputum eosinophils at the end of the study too. So once again, another piece of clinical data showing the pharmacodynamic effect of omalizumab. So some of the data from extra, going back to the study of our exploratory analysis of pheno and blood eosinophils. And what we do see, and kind of in line with the kinetics that I had mentioned in the previous study, where one of the mechanisms of action of omalizumab is the reduction of Fc epsilon receptor expression, is that we see a time-dependent reduction in pheno levels, while subtle, over time. And what you're looking at, the lower plots are the placebo-corrected changes, where at 16 weeks we see up to 15% decrease in pheno. Similarly, we see patterns for blood eosinophils, and as you can see, there's differences as in terms of the sampling based on the schedule assessments of the study. I also have to admit to you that we thoroughly investigated many of the type 2 biomarkers in the study, such as MCP4 and TARC that we reported as in terms of the context of our lebrikizumab program, and we haven't seen any effects on there. So we think that primarily the effects of omalizumab on eosinophilic biology. Okay, so now, let's check how we're doing on time. Some more recent data around our eculizumab program. So for those of you who are familiar with our eculizumab program in the past, is that the therapeutic hypothesis of this molecule was the depletion of M1 prime positive B cells. M1 prime positive B cells, they don't have their M1 segments genetically rearranged out, so these B cells express membrane-bound IgE. And we believe that the therapeutic hypothesis was that the depletion of these cells would prevent new allergic sensitization because it would interfere with differentiation of these M1 prime B cells to short-lived IgE plasma cells or differentiation of IgE memory cells. And also potentially it might reset the repertoire, therefore desensitizing subjects to allergens in which they've been sensitized to if we can deplete these B cells. So getting back to what I'm thinking about as a biomarker is the inhaled allergen challenge study. And this is data that you might be familiar with. And if you recall the omalizumab study data, while we did see an effect on the LAR as in terms of the inhaled allergen challenge study, the magnitude of the effect was not as great as with omalizumab. But what convinced us at the time about the activity of our molecule was really the biomarker analysis. And for me, because I'm involved in a lot of preclinical experiments as well, was some of this data where after one of the inhaled allergen challenges, we can see an increase in total IgE, which seems to be prevented by by treatment with Omalizumab, where there's approximately a 40% increase in total IgE. And this represents to me a classic memory B-cell recall response. And also more sensitively, if we look at challenge-specific IgE, so subjects, they were screened on the basis of the panel of allergens, and they were and for which allergen which provoked an FEV1 decrease, they were subsequently challenged with the same specific IgE. We can see nearly a 10-fold increase on study as in terms of challenge-specific IgE, and this appeared to be prevented by Omalizumab. So at this time, we believed that we had an active molecule. When we did our phase 2b, it was actually quite disappointing for those of you who knew the results. There was a rather large phase 2b where there's approximately 540 subjects randomized 1:1, 1:1. And so we have 3 different active exposures. 150 mg quarterly, 400 mg quarterly, and at the highest exposure is 300 mg monthly. And what you're looking at the forest plot is the treatment effect, where going to the right, it favors Omalizumab versus placebo. And as you can see here is that we did not reach statistical significance. As in terms of clinical significance, we only achieved a 19.6% reduction in exacerbations, which was very disappointing. However, upon closer examination of the biomarker data, I think that there are some implications to it and can set up new hypotheses. However, they're untested hypotheses. If you look at the total IgE, and just to remind you, if I had neglected to mention before, the primary outcome was 36 weeks before termination of dosing. We see a steady decline in total IgE and allergen-specific IgE up to 36 weeks. However, it's a gradual decline. And if you look at the lower plots, these are the percent changes. So while we did not capture or we could not demonstrate that there was clinical efficacy in this, or sufficient clinical efficacy in this study, one might interpret or hypothesize that we had conducted a longer study where total and specific IgE was reduced for a longer period of time, as perhaps we would have observed sufficient clinical efficacy. And now I'm going to transition a bit to some unpublished data. So this slide, I want to provide some context to really kind of set up why we were interested in conducting this study. And I'll go into the data a little bit more in subsequent slides. We believe that mast cell localization, airway smooth muscle, is a defining pathophysiologic feature of asthma. And this is really based on work from Chris Breitling and Pete Brading at Leicester. As all of you know, mast cell tryptase is abundantly expressed by mast cells and, of course, by basophils. And we believe that its enzymatic activity can lead to remodeling effects, especially on airway smooth muscle, and also direct effects on airway smooth muscle that might lead to airway obstruction. And then finally, we describe loss of function mutations that are common among diverse ethnic population among the tryptase genes. So putting it all together, really we had a question a couple of years ago. How do tryptase loss of function mutations affect tryptase activity and expression? And in the case of our reverse translational work, how does this affect the clinical responses to anti-IgE therapy, since ostensibly we are targeting mast cell biology? So just to review some of the data, in terms of what calibrates some of our assumptions, this is work from Chris Breitling, where he did an immunohistochemical analysis of the pathology of asthmatic airways, where on the y-axis we have the number of mast cells and airway smooth muscle bundles. On the x-axis we have normal controls, subjects primarily with mild asthma, and subjects with eosinophilic airway inflammation but without airway obstruction, so eosinophilic bronchitis. And what they concluded from this study is that there's an elevation of mast cells, specifically in airway smooth muscle bundles, in subjects with asthma. They later followed up this work and showed that there was a direct correlation with the number of mast cells in airway smooth muscle bundle with airway hyperactivity. And in this case, and they didn't focus on methylcholine PC20, for those of you who don't know what that is, it's provocative concentration to elicit a 20% decrease in FEV1. They furthermore have shown that it was in particular these Thy1 positives, so these fibroblastoid-like mast cells that have seemed to correlate very well with airway hyperreactivity. Further linking mast cell biology with airway hyperreactivity, I want to point your attention to this study from Cahill et al. It was a study of imatinib, which is a tyrosine kinase, which among the number of signaling molecules that it inhibits KIT signaling. And KIT signaling is important as in terms of the survival of mast cells. What you're looking at here in the study was a relatively small study of, I believe, approximately 80 patients randomized 1-on-1 imatinib versus placebo. And what you're seeing is on the y-axis is the airway hyperreactivity measured by methylcholine PC20. And hopefully what you notice is that they observed a statistically significant improvement in airway hyperreactivity, presumably by affecting mast cell survival in these asthma patients, further linking mast cell biology to airway hyperreactivity, one of the cardinal features of asthma. As in terms of the biomarker analysis, what I was really pleased to see was that they reported a reduction of total levels of tryptase in the periphery. So we would expect that with depletion of mast cells in asthma subjects that we would see a commensurate decrease as in terms of the production of mediators in which they produce. As in terms of what do we think is the biological consequence of tryptase, and if I had to mention to you, for those of you who don't know, tryptase is a serine protease, and one of the particular effects that we think it leads to is airway muscle contraction. This is some other work from the University of Leicester, where they're looking at airway muscle cells embedded in collagen gels, and they are treated with exogenously with beta tryptase. It's the active isoform of tryptase, as well with tryptase and a protease inhibitor, the peptin, to really get at the question about the dependency of this outcome with respect to the protease function. And hopefully what you can see is that there is definitely a time-dependent effect as in terms of airway smooth muscle contraction in vitro with exogenous tryptase. And really, this is some of the data that leads to the hypothesis that tryptase might be important as in terms of affecting airway smooth muscle tone and therefore airway obstruction and airway reactivity. There is a lot of work as in terms of the literature that has described potentially how mast cells and its products may affect different aspects of asthma biology, one of it being neuroinflammation. And so primarily the focus that I'm going to be talking about today is as in terms of its protease activity on airway smooth muscle cells. So getting at the pharmacogenetic effect, and I'm going to take my time talking about this in case you're not familiar as in terms of the genetics of the tryptases. So on chromosome 16, and what I'm showing you here is a figure of 4 tryptases. So gamma, TPSB2, TPSAB1, and delta. So one of the problems about this gene cluster is that they're highly homologous, and it's problematic for someone like me who's interested in whole genome sequencing is that they multi-map. So when we do short read alignments, really the alignment program is really obfuscated by this high degree of homology, and so the tryptase cluster is largely ignored by modern day So what I want to focus on is some of the things that we do know about it. And in particular, it's about TPSB2 and TPSAB1, which has the ability to express dissoluble tryptases. And the loss of function mutations that I mentioned at the beginning really are described here, whereas in terms of TPSB2, there could be a beta-3 frameshift mutation, and at TPSAB1, there's an alpha isoform. As in terms of what underlies these mutations, for TPSB2, the beta-3 frameshift is associated with a 109 residue truncation spanning the catalytic domain. So we believe that this is actually a functional allele. For TPSAB1, there are multiple amino acid changes, mutations. 2 of them that I think are especially important. One of them at the propeptide domain, which in vitro prevents autoprocessing of tryptase, as well as one at the catalytic interface that affects its ability to interact with its substrate. So in the way that I think about it, there's at least 2 different hits in which affect the function of alpha. And the way that I'm going to present the data to you is kind of a simplified way, is that you can think about in a particular individual, we can sum up the number of active tryptase alleles. So you can get a gene dosage of the number of active tryptase isoforms, genetic isoforms in which they are able to express. And I'm calling this active tryptase allele count. So we want to better characterize what is the consequence of this. So what this experiment tries to capture is, on an individual mast cell basis, does genotype actually affect tryptase activity? So to get at this question, we isolated primary mast cells from foreskin donors, and lysate tryptase activity was assessed by a substrate activity assay. And this was in the presence of alpha-1 antitrypsin. And something I didn't mention to you is that tryptase has no known endogenous protease inhibitor. And so we did this in the presence of alpha-1 antitrypsin to inhibit all other, that we know of, proteases in the lysates. And hopefully what we're looking at on the y-axis is a reflection of the tryptase-specific activity. So in the non-KIT selected cells from these primary cells, we genotyped them, and what we see on the x-axis here is active tryptase allele count. And what we see on an individual mast cell basis is a correlation of tryptase activity with genotype. So what we concluded from this study is that active tryptase allele count is a determinant of the degree of activity of mast cell tryptase. So now I want to transition into in vivo clinical data. So I mentioned to you before that the high homology of the tryptase gene cluster confounds a lot of conventional genetic analyses. And what we had done in this particular study is we've taken traditional PCR Sanger sequencing to evaluate this gene cluster a little more thoroughly in some of our asthma, severe asthma patients. And in this case, in particular, we selected European ancestry subjects. We determined this based on conventional genotyping arrays, and we did PCR Sanger sequencing. One of the things I want to mention, though we are not powered for it, is that we don't really have at this time a signal for association or risk with active tryptase allele count. And also, I just wanted to show you the distribution of what we observed. That primarily, subjects having 1 active tryptase allele count are very rare, and we have not observed anyone with having 0. And this probably has to do with linkage disequilibrium. And subjects having 4 active tryptase alleles are relatively rare as well. And that predominantly most of the subjects have either 2 or 3 active tryptase alleles. So the next question we wanted to ask, in severe asthma patients, how does active tryptase allele count affect their expression of tryptase in asthmatics? So what we're looking at is baseline samples from one of our severe asthma studies called the Millie study. And I can provide more details if necessary. And what we've noticed is that we saw a distinct and actually a very impressive PQTL, so protein quantitative trait loci, with active tryptase allele count and serum total tryptase. And we were able to replicate this in another study, Bobcat, which I mentioned to you before, where we see in the plasma, we see a significant PQTL as well. And also I should mention that we've actually found this in healthy controls as well, though the slope is not as steep as you can see here, where in this case, in Millie, we see an approximately a 2-fold increase as in terms of total tryptase for subjects with 4 allele counts versus 2. So this is very satisfying that we see a direct correlation with gene dosage with the expression of total tryptase. So now getting to the pharmacogenetic analysis that I had mentioned to you about this before. Once again, I'm going back to the EXTRA study, so the study of Omalizumab. So it was a randomized controlled study of Omalizumab in atopic severe asthma. And this is the data as in terms of the distribution of active tryptase allele count. As you can see here, for subjects who have 1 or 4, it's a very, very small group. So in subsequent analyses, I'm actually just dichotomizing this population for subjects who have either 1 or 2, or 3 or 4. We conducted PCR Sanger sequencing, and these are subjects of European ancestry who are based on their admixture coefficients. They're greater than 80% European, and which was a subset of the larger study. So in total, we had 309 subjects. And at baseline, these are some of their characteristics as in terms of their clinical characteristics, as well as their biomarker characteristics. And so this is really to kind of satisfy the question is that whether or not there was some differences at baseline kind of affected their clinical benefit at the end of the study. And if you can look here, in general, I think that they're quite comparable for all of these measures. And where we do see a pharmacogenetic effect is with respect to lung function improvement. Omalizumab has been described to have a rather modest impact on look on the left, and first of all, on the y-axis is the percent change in terms of FEV1. The x-axis is as in terms of weeks. It's a 48-week study. And hopefully, what you can see here was that there's a distinct benefit treatment effect difference for subjects who were active tryptase allele 1 or 2 versus 3 or 4. We also looked at exacerbations, but also getting to the point, since we only looked at 309 subjects, we could not conclude that there was a pharmacogenetic effect on exacerbations, and perhaps it's because of insufficient statistical power. But what we think this data has suggested is that using active tryptase allele count identifies a population of asthmatics who not only benefit from in terms of exacerbations based on omalizumab, they also derive improvements in lung function. So I mentioned to you before that type 2 inflammation and the biomarkers which measures type 2 inflammation, periosteum, pheno, and blood eosinophils, have been used in clinical development programs for therapies targeting type 2 inflammation. So one of the questions that we asked, what is the relationship between active tryptase allele count and what we presume to be tryptase function, so is serum protease activity with type 2 biology? And looking at a culmination of data from Bobcat, Extra, and Millie, we're looking at the biomarker analysis for serum periosteum, pheno, and blood eosinophils. And if you could just ignore the subjects, which were 1 or 3 or 2 subjects in each of these studies, having 1 active tryptase allele count, train your eyes to subjects having 2, 3, or 4. We see that levels are comparable across the spectrum of tryptase allele count and presumably intrinsic ability, serum protease activity in these particular subjects. Which brings us, especially from a perspective of someone working on new therapies for asthma, is that potentially subjects who have low levels of these type 2 biomarkers, which many of the new novel therapies that are coming to market nowadays, such as mepolizumab and dupilumab, primarily aren't thought to have a great impact. These are the subjects with elevated active tryptase allele count and presumably elevated levels of active tryptase protein, suggesting to us that perhaps if you were to target this biology in these particular subjects, that this might satisfy an unmet medical need. So how do we interpret all this data? And I just want to remind you that there's a number of ways mast cells can be elicited as in terms of the secretion of soluble mediators. There's definitely epsilon receptor IgA-mediated cross-linking, which all of you are familiar with. But there's also GPCR stimulation, such as through substance P, and also cytokine signaling that may induce the activation and secretion of products from mast cells. So with this in mind, we have a speculative model. And hopefully what I convince you today is that active tryptase allele count is a determinant of intrinsic active tryptase expression. So if we think about secreted active tryptase, some of it is dependent on Fc epsilon receptor or IgE-mediated mechanism, which is kind of represented as cartoon by gray. And if we were to assume after the context of anti-IgE therapy, is that perhaps in individual subjects, is that we might reduce their level of secreted active tryptase in these patients. And in the subjects having low levels of active tryptase allele count, these are the subjects who are brought down to an important clinical threshold. And in these subjects, they derive enhanced clinical effect. So in conclusion, in the recent past, based on the omalizumab experience, we believe that the omalizumab data has informed clinical development decisions for omalizumab. Unfortunately, the phase 2b outcomes weren't what we had anticipated, but we believe that we have made decisions based on its molecular activity. And also more recently, in some of the unpublished data that I had just presented to you, is we've done analyses of the pharmacogenetics of omalizumab, where primarily active tryptase allele count correlates with mast cell tryptase ex vivo. So on an individual mast cell basis, it determines tryptase activity, the total circulating levels in severe asthmatics, and treatment responses with respect to omalizumab. So one of the major caveats for some of these analyses are the post hoc nature of these analyses, and we're looking for replication studies as we speak. So the implications of this work is that we're very interested in mast cell tryptase, and that mast cell tryptase activity we hypothesize contributes to airway dysfunction in asthma, and that we believe tryptase genetics may be useful as in terms of an instrument to understand and interpret potentially studies going forward. So some of the future work that I'm involved with is developing algorithms to call tryptase alleles based on short-read whole genome sequencing data, where we can really harness some of the data that we have in our Genentech cohorts, and also evaluate this pharmacogenetic hypotheses in other studies. As all of you know, is that Omalizumab has been approved in multiple indications, and I'm very interested to see if some of these pharmacogenetic hypotheses pans out in those other indications as well. And finally, I want to acknowledge my colleagues who have worked on many of the studies that I had mentioned to you before. And I want to call out especially Choy at Immunology Discovery, and also the Clinical Development Group. And thank you for your attention.
So you sort of addressed this question in your last figure slide there, but why would having fewer active tryptase genes make you respond better to Omalizumab? And I take your model here, but then wouldn't that suggest if you up the dose of Omalizumab or something else, you could affect? I mean, it's counterintuitive. You think the more active tryptase genes you have, the more likely you should be to respond to Omalizumab.
Yeah, just to, sorry if I didn't emphasize this enough. As I kind of mentioned, there's a lot of different ways that tryptase can be released, in addition to IgE-mediated pathways. And especially for those subjects who have a predisposition to express more of these potentially pathogenic mechanisms that may not be ameliorated by current therapy, I think those are the subjects that wider have less clinical response. Also want to mention that this was specific to airway function, so airway obstruction. And while I have to point out the caveats about exacerbations that perhaps were underpowered, directionally we didn't see a difference, at least with respect to that outcome. And I think what this work kind of showed is that there's a subpopulation that actually derives meaningful improvements in terms of airway function. So I hope that addresses your question.
Are there any models where you eliminate tryptase, like a monoclonal antibody against tryptase itself? Has that been studied, or is that too dangerous?
Years from now.
So there's a speculation that that's a model target that we could utilize in the future?
So I think it's a good target.
Okay. Corporate standing. Okay.
All right. Well, I also want to mention to you, I mean, there was a small molecule inhibitor, and it was conducted in the, both in a sheep and human inhaled allergen challenge. I think it was APC 366. And this particular molecule was effective as in terms of the LAR responses. That molecule, for many different reasons, and it's not a Genentech molecule, wasn't developed any further. So it is thought that, in the context of what we think to be possible surrogates of a response to asthma, is that tryptase inhibitors can be effective.
I was wondering if in the subjects that experienced attenuated early phase responses in response to Omalizumab, any biopsy data there evaluating mast cell density, activation, post-allergen challenge, the percentage activated?
Yeah, unfortunately, there was never a bronchoscopic study conducted, so a mechanistic study was never conducted with Omalizumab. As a researcher, I would have loved to see that, and also, I mean, there was a lot of other assessments that we didn't include in the publications, where we actually looked at bone marrow by flow cytometry, because we were looking for these M1 positive cells, and we're looking for pharmacodynamic effects on these. But a lot of these didn't pan out for either technical reasons or perhaps not enough drug activity.
For the new molecule that you showed about depleting B cells for IgE, I didn't see like any safety data because that will, protect for parasitic infections. There were some data, and that will be like something feasible in the future, because that's like shutting down another protective way of the immune system. So I don't know.
Yeah, definitely. So what I can say is in the Omalizumab program, this has already been published, that it was well tolerated, having been exposed to, I think, over 1,000 subjects, including over 600 severe asthma subjects. It's well tolerated. and and AEs were relatively well balanced between placebo and active treatment. So we think it's it's a safe treatment paradigm. I know that there's been some off-label use of Rituxan, so it's anti-CD20 in the context of asthma. While as in terms of signals of efficacy, I'm really unconvinced whether or not they saw a signal. And so I don't think that they actually reported significant AEs in those studies, at least the ones I'm aware of. So, but your caution or your concern is well understood. Thank you. Yes.
One thing you didn't talk about at all, and I'm curious if it would inform this, is the efficacy of omalizumab for chronic urticaria. And I asked specifically in regard to the tryptase allele count. I mean, we now know that higher tryptase alleles are seen in mast cell disorders. I don't know that anybody's looked at that in chronic urticaria, but if you think of it as a spectrum, does that help us understand?
Yeah, so we're working with Josh Milner
Yeah.
on that, and so, also just provide some background on that. specifically what he described were the copy number variants.
Right.
And so the copy number variants, these were duplications and triplications of the TPS-AB2, and we believe that those were the alpha genetic isoforms at that loci. He's shown that work a couple of years ago with John in Nature Genetics.
So is that a distinct metric from what you're saying?
It's a distinct metric. And so that seems to occur in about 5% of Europeans, and it's less common in other ethnic ancestries as well. And to kind of jump ahead about the whole genome sequencing methods, algorithms, we're actually pretty close to finalizing. We've looked at this, and it doesn't seem to affect clinical responses, but it affects circulating tryptase levels. Any other questions? No, thank you. Put in your phone bag. You still